Hi everyone, this is Ratna. Welcome to Basics of Biotechnology. In this tutorial, I am going to discuss about central dogma. Do you know what exactly the central dogma is? It is a process by which the instructions of DNA are converted into functional products. See, DNA is converting into RNA and RNA is converting into proteins. The replication, transcription, and translation are the three main processes used by cells to maintain the genetic information and to convert the genetic information encoded in DNA into gene products. As I already told you, transcription means converting DNA into RNA. See, it is a process, the first part of the central dogma of molecular biology. DNA is converting into RNA. It is the transfer of genetic instructions of DNA to messenger RNA. See, during transcription, a strand of mRNA is made that is complementary to the strand of DNA. See, it is the mRNA strand and which is complementary to the strand of DNA. There are three steps involved in transcription. First one is initiation and second one is elongation and third one is termination. In the primary step of initiation, what happens is the RNA polymerase binds to the region of gene called promoter site. See, this is the promoter site and this is the termination site. This signals the DNA to unwind so that enzyme, RNA polymerase enzyme, what happens, it can read the basis of the DNA strand, this template strand. So it will read the basis of the template strand and makes the complementary sequence of basis of this template strand. See, RNA is made by copying the information from the template strand of DNA and it is synthesized in a 5 to 3 prime direction, just like DNA. See, DNA template strand, it is making the copy based on the template strand, and this template strand, it is in 3 to 5 prime direction, so that RNA will form in 5 to 3 prime direction. What is data box? Data box is a DNA sequence that indicates where genetic sequence can read and decode. It is a type of promoter sequence which specifies to other molecules where transcription begins, and it also able to define direction of the transcription. See, it is a starting point of the transcription, and what happens is the RNA polymerase binds to the region of this promoter site, this Tata box site, and this signals the DNA to unwind. See, the unwind has happened at this region, and so the enzyme, RNA polymerase enzyme, can read the bases in one of the DNA strand. This RNA polymerase will read the basis of this template strand. Now the RNA polymerase enzyme is now ready to make a strand of mRNA. See, it is now ready to make a strand of mRNA with a complementary sequences of bases. See, with the complementary sequence of bases. In initiation step, what happens is RNA polymerase bind to the promoter site and this signals the DNA to unwind so that enzyme can read the bases in one of the DNA strand, nothing but template strand, and this RNA polymerase is now ready to make a strand of mRNA. See, it is unwound, it unwounds the DNA and it is now ready to make a strand of mRNA which is complementary, this which is complementary to with a sequence of bases to this DNA template strand. And now in elongation, what happens is the newly formed RNA is bound to the unwound. See, this newly formed RNA bounded to the template strand, and after this elongation, what happens? This completion of RNA transcript is completed, and what happens is this termination will happen if the RNA polymerase crosses this termination site. See, this is the promoter site and this termination site. RNA polymerase attaches to the promoter site at the time of initiation, and if this RNA polymerase crosses this termination site, then termination will occur and the mRNA strand is complete and it ditches from the DNA. What we need for transcription initiation? We need a promoter site, the start of the gene to be recognized by the promoter site, and we need a DNA template, which is opposite to the actual sequence of the gene, and it is in 3 to 5 prime direction. And RNA polymerase enzyme, RNA polymerase 1, 2, and 3, which depends on the gene and the regulatory elements, loop over and interact with the promoter and controls the amount of RNA being made. In this elongation of transcription, what happens is RNA polymerase starts making the new RNA strand. Elongation is it is the addition of nucleotides to the mRNA strand. RNA polymerase reads the unbound DNA strand and builds the mRNA molecules using complementary base pairs. See, it moves from the promoter and it leaves the initiation transcription factors behind, and it gains some elongation factors to elongate the transcription. And RNA polymerase generates a single stranded RNA, only single stranded RNA. This creates a transcription bubbles which moves along the DNA template, and the RNA strand only base pairs with the DNA template strand for only 8 nucleotides only. After this, the DNA becomes double stranded again. Transcription termination is the end of transcription, and it occurs when this RNA polymerase bubble crosses the stop sequence, this termination sequence of the gene. The mRNA strand is complete and ditches the DNA. See, elongation will continue until this RNA polymerase encounters the stop sequence. At this point, transcription stops, and the RNA polymerase release the DNA template, and RNA will form and DNA back to its original position.